today, our program is, I'll go through a little introduction of virtual screening, what we mean by virtual screening, and we'll gear our presentation towards a virtual screening pipeline that me and my collaborators together, Travis Wheeler, Vish, and others have developed. So the talk will be around that. And after my talk, Vish will have a tutorial walking all of you through the some part of the pipeline. Now, what do we mean by virtual screening? So typically, a drug development project starts, there is a lot of biology goes when you have a disease or some kind of malfunction in your cell. A lot of biology goes to identify the target. That means like identify the protein or proteins that is causing the malfunction. And that's years of work. But once you have identified that and you want to rectify it, then comes what kind of small molecules, nowadays it can be peptide as well, what kind of molecules we can design or identify so that most possibly we want to stop the protein that's malfunctioning from its natural activity. And that is called generation of hit. That means like you are trying to identify what molecules can interact with the protein of interest. And after the generation of hit, you have some initial idea, then you want to optimize it further because your initial hit that you generate, initial molecule that you identified, may not be optically interacting with your target. And that's the part called drug development. And Sudeep is going to focus on that part, the computational aspect of that tomorrow afternoon. And then once you have identified what we call a lead, then you try animal model and finally a clinical trial. Now, usually it takes 10 years, on average, more than 10 years to run through the whole process. And the initial part is mainly done by the academic, and the later part is mainly done by the pharmaceutical industry. And on average, you have more than $2 billion to develop a drug. Now, in the first part, when you have identified the target, at least a few years back, before the computational field developed, typically the big pharmaceutical industries, what they used to do, they have their own in-house compound libraries where they have millions of compound has been synthesized. And then you have your target, you create an assay where you blast through all these millions of compound and see which one binds. That's called high throughput screening. It's an experimental screening. And then from then on, you follow the next steps, typically of the order 1 in 5,000, of the compound library that typically binds to the target. Now, the part that we can help computationally is to accelerate that part where experimentally you have to synthesize 1 million of compound and create so many assays and do a high throughput screening. At least that part we can accelerate computationally. And when I'm saying accelerating, that means like we are reducing the burden on the experimental side. We are reducing the burden on the assays and so on. So in an experimental, a typical experimental setup, if you start with, let's say, 100,000 compound you have to synthesize, you will end up roughly 125 hits. These are all on average numbers, but not all the 125 hits will be super unique molecules. Like, they'll be very similar molecules. And ultimately, when you're looking for different kind of molecules, you probably be ending up with 5 to 10 different unique scaffolds. That means like molecules that are structurally different from each other. And this cycle can take few months to up to 2 years. And this is the part that we are trying to accelerate. Now, keep in mind, we are not trying to replace it. Computational methodology are still not accurate enough to replace an experimental assay. What we are trying to do is we are trying to reduce the burden on the experimental side. That means instead of 1 million compounds, if we can computationally suggest an order of thousands of compounds and come up with the same number of hits, then we reduce the price by a factor of 1,000. So that is the goal of the computational aspect. So again, this slide was prepared for SARS-CoV-2, but the initial part for you is identify the target. So whatever disease or protein you are working with, it's good to know, it's good to have a clear idea which protein you are going to target and why you are targeting it, because just targeting any protein, create a drug, may not work, in the sense that you want to target the protein before it enters the cell of the human cell, or you want to target it after it enters the human cell, and so on. All those things we have to take into consideration. Like in case of SARS-CoV-2, the spike proteins are actually what is the target of the vaccine. One of the reasons being is like that's the entry point. You don't have to go inside the human cell. You can attack them outside the human cell. But the drug remdesivir, on the other hand, attacks the protein inside the human cell. So those details you have to understand first, because just targeting any protein is not going to help. Your drug may not reach the target. There's a lots of biology goes behind it, okay? So whenever you are working with a target, make sure you understand the life cycle of that pathogen. This is the life cycle of SARS-CoV-2. On the left corner, you can see how it's entering the cell with the spike protein binding to ACE2 receptor, and then the RNA is getting released and multiplied and repacking and releasing. Now, for your protein, for your pathogen, you have to understand the life cycle better. Again, this is because all of you are starting, your research project. You have to be very clear which protein you are targeting and why you are targeting and which part of the life cycle you are targeting. Otherwise, the whole effort might be futile. So, again, this is a case of SARS-CoV-2. We targeted 3 of the proteins, and nucleocapsid, NSP12, and 3CL Pro, but for you, it will be different protein. So when you are targeting protein, one of the reasons drug development takes such a long time is the toxicity. The problem is that a toxicity you can only find when you go through the whole cycle. You develop a target, you drive the mammal model, you run the clinical studies, and then you are finding, oh, this drug is toxic to the human. You have to come back and start again. Some of the ways you can avoid having too toxic is if you target a protein, it's especially for bacteria, if you target a protein that does not have many mammalian homologs, then you kind of have an idea, okay, whatever I design against it is not going to attack any other human protein, so on. So you can reduce the toxicity by that. And then any initial part of the drug development requires a good structure, initial structure, because from now on, what we'll be doing requires the protein structure. So either your protein has to have a good structure, salt, or a good model using probably AlphaFold 2, which you will have later part of the lecture, in the next weeks, we'll have some session on AlphaFold 2. So make sure your protein has a good model, because a lot of the subsequent step depends on how well we know the protein structure. So that is a very good initial requirement, that you have a good modeling. And then another part that we almost always tend to forget, actually I used to forget when I was learning all this stuff, is glycosylation. Depending on where the protein is, your protein might be highly glycosylated. And then the glycosylation modeling is just maturing now. I mean, we can model the glycosylation, but it's not, it's an evolving field. So if you're targeting a protein, if it has the possibility of glycosylation, like the spike protein of SARS-CoV-2, make sure you model the the sugars there, so that you know which part will be exposed and so on. Or if you can find a protein that does not require glycosylation. So glycosylation means like the protein has all this amino acid, and the body has this sugar. Some of the sugar binds to the amino acid. If you look at the spike protein structure, the real structure, you'll see a lot of sugar is covering the spike protein. You almost cannot see the surface of the spike protein. So those things you have to keep in mind. So let's say we found our target. Okay? We created our model. We have a good protein structure. The next part, remember, the pharmaceutical industries have typically 1 million of molecules they have synthesized. Over last 10 years and so on, since the zinc 15 came online, we have access to billions of synthesizable molecules that you can calculate computationally. Okay, so just think about the order. Pharmaceutical industry can synthesize 1 million of molecules. It takes money to synthesize, okay? But we have virtually collected, and when you say we as a field, virtually has collected of the orders of billions of molecules that we can computationally screen. On top of that, most of this molecule is synthesizable. That means they're real. Like if you like one of the molecules, you send it to the vendor and say, like, can you please synthesize it to me? So in this list, we have created, and this database is available. Vish will go through it later. In this list, what we did is like, we collected all the publicly available database known to us. There are some more maybe. And then we removed any duplication that was done by Travis. He removed all the duplication. So we have collected a 3.7 billion unique molecules that you can use for screening any of your target. Okay. Now, the small molecule world is really large. There are so many possibilities. So 3.7 billion is a good number, but it's still a fraction of the number of possible small molecules there can be. So now we have these a number of small molecules. We have identified the target. So what we want to find is like, which small molecule will interact with my protein, okay? So how do we calculate those? A lot of them, you are already interested in molecular dynamics. So molecular dynamics is one way to identify if my ligand will interact with my protein. But the problem is, MD simulation takes a lot of resources and a lot of time. If you have a reasonable number of ligands and a single target, you can run it by MD if you have access to high performance computing. But if we want to, let's say, run 3.7 billion of molecules, and let's say we are targeting 3 protein, and we have a possibility of 22 pockets. If I was given 5,000 CPUs, it would have taken me 1,200 years to complete all the MD simulation. So we are doing the initial screening where even though MD simulation is a more accurate descriptions on what's happening, but for such a huge number, we cannot actually use the MD simulation. It will come later part. If we want to be more accurate, we can go quantum mechanical. The problem with the quantum mechanics, there is a size limit, typically of the order of thousands of atoms. Your protein is more than thousands of atoms. So if you want even more accurate, the QM calculation is not a possibility. Now then docking, which a lot of you are interested in, is another possibility. But even when we are dealing with the billions of molecules, even docking takes a lot of time. Okay? So for this scenario, if I was given 5,000 CPUs, I would have needed 12 years to finish all the dockings of 2.2 billion molecules, of 3.7 billion molecules. So we needed a different approach, and docking is good when you have millions of molecules. But the universe of small molecules is billions, if not even more. So if you want to scan all these molecules, we need a different approach, how we can scan them. It's not that we can scan them exhaustively. We can scan them in a good, confident way so that we have an idea what are the initial hits, okay? So these are the tools like docking and molecular mechanics, like MD simulation, you will be using the later part of the drug screening. But we needed a different approach to deal with such a huge billions of molecules. And so this is an approach, I would say it's, there are different parts of approach, and I'll go through slowly. It's not unique in the sense that different people has done different parts, but it's unique in the sense that all of them has been put under the same umbrella as a single software pipeline. And then there are some innovations here too. So this pipeline, which we call drug sniffer, and we'll go through it in more details, allows you to screen through billions of molecules the reasonable number of HPC computational HPC resources available. Okay, so let's see. So the first two part is, again, target identification is the biology that you need to know. You need to collaborate with your biologist friends, or if you are a wet lab person, you need to know really well which protein you want to target. And this is step number one, but it may take years to identify that. Once you have identified that, the next part is modeling of the protein, and which has become easier and easier. So once that's like the first part, like modeling of the protein. Step two is like once we have a 3D structure, and then like yesterday, if you ran through the Chimera X tutorial, you saw how a ligand binds to a protein. So if you look at a protein, the protein has surfaces. Some of the surfaces is like a concave where something can go in and sit down there. And some of the surfaces are hydrophobic, where a hydrophobic ligand can go and sit down there. Those are called pockets. That means somewhere where the ligand can go and hide from the water. So in a cellular environment, you have water moving around all the time. Water is charged. So a ligand means like a small molecule or a peptide. It may not like the water, may want to go into the protein and sit down there. Those regions are called pocket. Now, identifying the pocket, like once you have a protein structure, How do you know which part the ligand is going to go bind? That is, again, an active field. We do not know for sure. Okay? The best way to know it is, of course, through experimental data. If you search the literature first. But if you do not have enough data in the literature, then we can do some prediction. And again, when you are using virtual screening, you have to remember this. You are approximating the real scenario through computation. That means the pockets that you are identifying, the region where a small molecule can bind, you are identifying may not always be real. Okay? So, but let's say you got a good idea, and this is where your knowledge about the field will come. So you got an idea like, where is my ligand is going to bind? Where is my small molecule is going to bind? So now we have a database of 3.7 billion molecules, right? We cannot search exhaustively that many molecules against that pocket. So what we do, and different people does different thing, we do is like, we create a de novo molecule, and I'll explain a little bit. And later we have a tutorial where you'll go through it. There will be explained more. A de novo molecule means we know the pocket. We try to attach different fragments of small molecules and try to grow a molecule that will fit to that pocket. That means I am not searching the exhaustive database. Rather, what I'm doing is I'm trying to get an idea what kind of molecule can fit into that pocket. And you will not get, all different kind of molecules that can fit in the pocket, but I can at least get an idea. And that's the call de novo design. That means we are designing some molecules from the scratch that will fit in that pocket. So for a typical pocket, you'll get of the order of a few thousands of such molecules. So once we get the order of a few thousands of such molecules, I'm going to skip this part. Vish can explain it later. Once you got a few thousands of molecules, then what you do, now we have out the order of a few thousands, right? We go back to our 3.7 billion or whatever billion database, and we ask the question, out of these initial molecules that found, how many of them, or how many of the 3.7 billion are similar to the one that we found? It's called a similarity search. So the idea behind it is, if two molecules are similar, and how do you define similar? We'll come to that. If two molecules are similar, small molecules, most probably they will bind to the same pocket. So that is the assumption behind it. So we take the thousands of de novo models, then we search that against our billions of molecules. And this is one of our innovations, that we can search these huge databases very quickly, the order of seconds or minutes. I don't remember the number. How long? A couple of minutes. So that's another of our innovation, that we can go through this huge database in a couple of minutes. So you take the initial seeds, which you call seed, that means like initially de novo design molecule, you search through the whole databases, and you're looking for similarity. Now, it's easier to say you are looking for similarity, but it's very hard to define how you define two molecules to be similar, okay? There are different methods, and we'll probably go to one or the two of the methods today, how you define two molecules to be similar. However, I must warn you, in terms of the similarity that we are looking, what we are defined by similarity is two molecules should bind to the same protein pocket. Or, in another term, two molecules should have the same activity. There still is not any well-developed definition that can identify two active molecules by any similarity search. So that is another area of active research in the sense that if you have a molecule, how do you define another molecule? How do you identify another molecule? How do you define similarity with another molecule that should have same activity? That means like those two should bind to the same pocket. That is an area of active research. We are still developing methods, but whatever the current method, we'll use it. So just remember, the similarity search, there are lots of assumptions in the sense that some of the current method will tell you two molecules are similar, but not necessarily they will be the active. So there is an assumption here. Now, we started with a few thousands. We ran through the, with whatever similarity algorithm we have right now, we'll end up with of the order of a few 100,000, okay? So out of the billion molecules, we identified now a few 100,000, which has the possibility of having the same activity against the same protein. Okay. So now we have reduced the order of the problem from a billion to a few 100,000. Now it's the order of, we reduced the order by a factor of 10,000. So now that we have reduced the order, now we can come back. Let's say, now we can come back and we can do the docking. So a lot of you are interested in docking. Aisha has been helping you with docking. So now we have reduced the problem of the billion molecules to a few 100,000 molecules. Now we can run the docking, okay? So you run the docking. After the docking, you'll get a docking score. Now, what does docking do? What docking does is like, it places the small molecule inside the pocket and then calculate different physical interactions. And the physical interactions is a combination of physical interactions that you get at the end. It's called a docking score. That means like the docking program is giving you the idea how good these two are matched. And in real life, what we want to measure is called binding affinity. That means like if I put the small molecule and my target protein in the solution, what is the probability the small molecule will find its way to the pocket? That's the real life experiment that we want to measure, called binding affinity. Now in a binding affinity, you have two types of interactions mainly called enthalpic and entropic. Enthalpic means like how the charges of the small molecules are interacting with the charges of the protein. Entropic means how the flexibility of the ligand and flexibility of the protein change the behavior of the binding. Okay. So in a docking experiment and any other molecular mechanics-based experiment, computational approaches, it's very easy to approximate the enthalpic part. That means like it's very easy to calculate the interaction between two charges. We know the physics, know the distance, we can calculate it. It is very hard to calculate the entropy part. The reason is entropy is you need to know all possible, like, if you go follow through the Boltzmann's equation and so on, you need to know all possible states the ligand can be, all possible states the protein can be, and then you have to calculate the probability and so on. So entropy is very hard to calculate. You can calculate some entropy through molecular mechanics, molecular dynamics. Not necessarily it is, you'll still get an accurate answer. So when you are docking, when you are doing the docking score, keep it in mind, you are reproducing mostly the enthalpic term. Entropic term will be approximated, okay? And there are different approximations, but none of them is perfect. And some docking score don't even include entropic term. So you got the docking score. So now we have a problem. My docking score may not correlate well with my binding affinity. And actually, it doesn't correlate well with binding affinity. Okay, especially when you're working with such a different, diverse molecules. I mean, docking is good when you are docking the similar kind of molecules into the protein, with slight changes, because similar molecules have similar entropy. But if you're docking huge number of different molecules, their entropies will be different, and docking score will not be able to take into account the entropy. So keep it in mind, your docking score will not reflect the binding affinity that you are trying to reproduce. Binding affinity is the experimental value that you are trying to reproduce. So that's where our next part comes in, which is like trying to develop, we called it an activity prediction because a lot of the experimental data, it is easier to get activity prediction experimental data that like IC50, EC50, that means like what is the concentration of the small molecules you have to put before the cells become inactive or something like that. So it's easier to get, that's called activity data. It's easier to get that data. But the binding affinity is a thermodynamic experiment where you have to put the two, purified protein, purified ligand, and measure the temperature change of the system when they bind. So that's the binding affinity. That's a harder experiment. So it's not easy to get all the binding affinity data. It's easier to get the activity prediction, which is like IC50, but they can be correlated. So here we are trying to develop in the drug sniffer. There is a model, and this model is an active development, where you input the different terms of the docking score, like a docking score. I do not know if you'll have time to go through it, but when whoever you are working in details with drug development, we go through it. In a docking score, you have electrostatic interaction, electrostatic repulsion, van der Waal repulsion, van der Waal interaction, and those terms. Van der Waal repulsions mean two atoms cannot be at the same place. Van der Waal attractions means screened electrostatic and so on from the nucleus and so on. There are different terms. So instead of a single docking score, we extract all those different terms, and then we add some other terms with it, which we think will mimic the entropy. We have a specific model of entropy as well, which has not been incorporated in this one yet. But currently, we have some other approximation, not so good approximation for entropy. And we have developed a neural network model to predict the activity score. Now, this model is as good as any other model currently in the field. However, it's still not good enough, in the sense that roughly, you'll get, I don't know, the AUC is a term like how much true positive, that means how much real ligand you are identifying versus how much false ligands you are identifying. So AUC of that one is of the order of 0.7, which sounds good, but when you are screening billions of molecules or millions of molecules, you still end up with lots of false positives. So this is another area of active development we are working on. We'll skip through that. So that's, if somebody is interested in the details of the neural network model that Daniel has worked, this is the detail, and you can talk to Daniel, because one of you, whoever you are interested, would like to develop it even further. Okay. So this just shows how our method compares to other current methods in the market, and we are the best. So, okay, the final part is after you have identified which molecules are possibly binding your protein. Remember, I told you the major bottleneck of drug development is toxicity. The reason is like it is very hard to model toxicity. And then that's where a lot of the drug fails in the clinical trial. It takes a lot of money to go to clinical trial. So if your toxicity fails, you have to start back again and so on. So we have this another area we are working on, which is like, can we filter some of the obvious toxic molecules from our list? So that's the last part of our pipeline. So if you, and this pipeline is a very general in the sense that you can bring any of your target and run through this. This is very modular in the sense that instead of running through the whole part, you can run individual part on it. Let's say you do not like our activity prediction neural network model. You want to bring your own model, you can plug it in there. Let's say you want to use some other similarity search, or you want to bypass docking. You want to run all MD simulations and then created the docked model. You can do that here, very modular. At the end, what we are expecting is that we'll give a list of thousands of molecules, maybe a few thousands, to our experimentalist friend and say like, here, take this 1,000 or few thousand. I guarantee you, you are going to get 100 unique hits. So remember, initially, an experiment has to synthesize millions of molecules to get 100 unique hits. With this process, we hope to reduce that number and say like, synthesize a few thousand, and hopefully you'll get 100 hits. So again, as a field, we are not there yet to confidently say this molecule, we predicted this molecule, and this is going to bind to the target. Because there are so many assumptions that goes inside that every step. So as a field, we are not there yet, but we are improving. But what we can do is like, we can reduce the burden on the experimental or experimentalist friends. We can create collaboration so that the drug development program that was beyond a small group before, there was only the big pharmas can do it, we can do it too. Okay, I'm going to stop here and I'll stop for, I'm happy with what I was able to communicate, but I'll stop for any question before I hand it over to Bish. So any question? Yes. Yes. You need to do a little bit of your own programming. This is our first release, okay? So in this release, you can change the sequence if you know a little bit of coding. Hopefully, in the future releases, we'll make the changes possible easier for the user. But yes, the answer is yes. You can do the change in the sequence. Yeah.
So my question is that I know something else as well, like that is called the pharmacophore modeling, and that is basic. We can also do it structure-based or the ligand-based. But what I want to ask that between these two methods, what should we prefer? Like a de novo design for of molecules according to the protein pocket, or just we should go towards the pharmacophore modeling? Because what I hear that is if we go to the, if we know the structure of that protein, so we can design much of the similar ligands according to the pocket, according to the properties of the pocket.
So what is Atiya's question is like, if we know the pocket and we know the pocket properties, that mean electrostatic charges and so on, and there are methods called pharmacophore methods. Can we design a ligand knowing the properties of the pocket and just go from on there on instead of doing all this search? And we have a recent paper coming out. Maybe is it on the bioarchive or maybe not? The fingerprint? No, it's not. Okay, it'll come out pretty soon. So the problem with those methods is to generate what we call different kind of ligands. This is called like scaffold hopping, in the sense that you need to generate, because we still do not know if you work with 1 or 2 or 10 ligands, whether they will be really active at the experiment. So we want to capture as many different kind of varieties of ligand with different unique structures. A drawback of the pharmacophore modeling is like to generate all those different varieties of ligand. So it is one way, but it is not as exhaustive way. And we are actually showing in our next paper how those kind of similarity searches misses the point identifying the active. And Vish, do you want to elaborate on that? Okay, so Vish is our specialist. He's going to answer this one on later. So the idea behind it is all those similarity search, we call it similarity because you have a property, you're trying to find this. They do not generate enough varieties of ligand. So that's the limitation we are trying to overcome with that. Yes. Yeah, so how do we handle the entropy? In our current model, which is not very good. So when you are calculating interaction between a ligand and a protein, the one way to calculate is calculate the physics term explicitly, which will miss the entropy. There is another way which is called statistical potential. Statistical potential means like you forget about the physics terms, you look at in your database and see like how often these two atoms are close, and how in PDB bind database and so on, how often these two atoms are close, and then if they're close very often, then they will get a higher statistical potential value. If they are random, then they will not get it. So that's another way of calculating docking score. One is the physics-based, one is the statistical potential-based. Now, statistical potential-based depends on the database, like how many bound ligand form you have, with the assumption that because we are not explicitly calculating individual forces, the entropic effect should be inside the statistical potential. And one of those potential is called d-fire, which we have included in it. However, this does not take into account explicit term of entropy. So we are developing another, in our future release, we'll developing another method where we can look at the surface properties of the molecule. That means like how much it is curved, where the electrostatic density, and get an quick estimation of entropy. So that is a future development. That's what I would like to go, where there's an explicit term for entropy. Currently, we rely on the statistical potential. Okay, if those are all, I will give it to Vish to help you run through the virtual screening or drug sniffer pipeline. And then there will be 4 of us helping, Travis, me, Daniel, and Jeremiah. Where is Jeremiah? Okay, somewhere. He disappeared, right? So we'll be helping you. So this is a tutorial. So you'll be following Vish with different parts. And wherever you get stuck, we'll help you with that. Thank you, Vish.